and we'll do our best to answer them. With that, let's get started. And it is my true pleasure to welcome Alex Reuben, who has done truly amazing work that you're about to hear about. Alex, I'm going to turn it over to you.
All right, thank you very much, Alana. John, did you want to present first, or you're good with me starting?
No, Alex, you can fire away, and I can go after.
All right,
Yep.
Good stuff. All right, well, thank you guys for having me. I appreciate the support, and I'm excited to be here today. So I'm going to tell you a little bit about the work we've been doing to bring T-cell receptor engineering to the RET fusion positive landscape. So we've known for some time now that the immune system is actually critical to protecting us from cancer, and specifically T cells. This is an experiment where mice without T cells were exposed to a carcinogen, as were mice that do have these T cells. And what you can see is that the mice without T cells actually develop cancer much more quickly than the others. And this is actually because T cells are actually very effective at killing tumor cells. Here's a short video. Here in pink, you can see the T cells. Here in green are the tumor cells. And as the T cells kill the tumor cells, the membrane of the tumor cell becomes permeable, and that's what lets it take up the dye that's in the environment and turn it red. Here's another example here with, once again, T cells in pink and the tumor cell in red. So really, they're extremely effective at killing tumors, and that's why they've been such an important focus for us and others in trying to overcome some of the resistance caused by cancer. Now, the way that T cells see which cells are cancerous and which are not is through something that's called antigen presentation. And basically, every cell in your body, every nucleated cell in your body is displaying at all times antigens at its surface that reflect the state of that cell. So a normal or healthy cell is presenting a normal or healthy antigen at its surface. You know, tumor cells are presenting different types of tumor antigens at their surface, and a cell that's infected by a virus, for example, will be presenting viral antigens at its surface. And so in all of these cases, T cells are able to see these antigens and discriminate between healthy and infected cells. And the way they do this is through what's called the T cell receptor. Here's a representation of that T cell receptor. It has two chains. This is what is the MHC molecule that I was referring to that's presented on all of your cells. And you have this short peptide at the surface that the T cell receptor recognizes to discriminate between a target cell that has an antigen of interest, so an infected cell or a malignant cell, versus a cell that does not, a cell that has, you know, a healthy antigen that obviously you would want to avoid for, you know, reasons of autoimmunity. And what happens when T cells recognize their antigens, and what I showed you in that video, is that the T cell receptor recognizes the antigen, and then it leads to killing of the target cell through one of many mechanisms. Here's one of those mechanisms here, where basically the T cell creates these pores in the membrane of the tumor cell, and ultimately that leads to the destruction of that tumor cell. So for this reason, there have been different therapies that have been developed to you know, leverage the power of T cells to overcome cancer. And one of those is adoptive cell therapy, where basically you have the resection of a tumor here, surgically resected, and then the tumor is cut into very small fragments, and it's grown over a few weeks. And so at the end of that, you have an expansion of lymphocytes to about 10 to the 10 different lymphocytes, so tens of billions, that are then reinfused in the patient to get rid of any remaining disease and protect from the appearance of metastases. And actually earlier today, there was a new paper that came out in the New England Journal of Medicine that showed in melanoma that adoptive cell therapy here with TIL is actually superior to checkpoint blockade with ipilimumab. And you can see that not only do the patients respond better, but the patients that do respond have these prolonged responses, you know, 7 years out. So extremely impressive responses. Now, this has also been done in lung cancer. Here's a clinical trial that was performed a couple years ago at Moffitt Cancer Center. Each one of these lines represents a patient, and you can see that many patients do actually respond. This represents the size of their tumors. And so for many patients, the tumors start to shrink, including some in whom the tumors completely disappear. But many patients did have, you know, recurrence, and these orange diamonds here represent the occurrence of new lesions here. So it's not a therapy that works for everyone in lung cancer. And so in lung cancer, one of the reasons may be the context, right? You're looking very closely at a tumor here, You're looking at the immune cells that are present in that tumor, but that tumor is actually located within the lungs. And the lungs, as we know, are constantly exposed to the air we inhale, including different types of viruses and pathogens. And so there may be an immune response in the lungs that it's actually detrimental and different from the anti-tumor response that we're hoping to study. And so we've looked at this as of others, and here in this experiment, what we did was to look at PBMCs, which are basically the T cells that are in your blood, and compare them to the tumor-infiltrating lymphocytes and see how effective they were at recognizing non-tumor antigens. In this case, these were viral antigens. And what you can see is that the tumor-infiltrating lymphocytes are actually very reactive to viral antigens, contrary to what you would expect, and certainly contrary to what you'd hope for. And beyond that, you can see that the T cells, or rather lung tumor infiltrating lymphocytes, in addition to recognizing what's called a tumor antigen here, also recognize other viruses and even bacteria. Now, beyond this, one of the hurdles with lung cancer is that you're facing what's called tumor heterogeneity. And what this is that these tumors, each cell is so different from the other that just because you're able to eradicate one cell doesn't mean you're going to be able to eradicate them all. And so if you look at a tumor that's very, very homogeneous here, ultimately you technically just need to isolate or rather amplify an immune response against one antigen to eradicate that tumor. But as the tumor becomes more and more complex, as is illustrated here, the immune response that's required becomes more and more complex, which complicates things much more further. And ultimately, there have been studies that have shown that when you look at tumor infiltrating lymphocytes, there's only about 10% of them that are actually able to recognize and kill the tumor that they've infiltrated. So really 90% of the T cells that are found in tumors are not playing any direct role to killing that tumors. And obviously that can be a problem when you think of adoptive cell therapy, where you're actually resecting the tumors and amplifying and reinfusing those lymphocytes. Now, what that means is that there are several T cells that are found in these tumors. Here's a section from a melanoma tumor here that's shown, and each one of these red circles represents a T cell. So the presence of the T cells is really not the problem. The issue is the T cells that are there are not the right T cells, perhaps for some of the reasons that I explained to you previously. Maybe they're recognizing non-tumor antigens, maybe they're heterogeneous and they're not recognizing antigens that are found throughout the tumor. And so what that means is that you have a scenario, like the scenario that's shown here on the right, where you have the same amount of T cells as you would on the left, but the T cells are just not able to recognize, to see the tumor and to destroy it here. In both cases, you have the same amount of T cells, but what you see here on the left is that as the T cells in red swarm around the tumor cells in green, they're able to recognize and kill those tumors. The tumor cells shrivel up and they detach from the plastic, whereas on the right, although you have the same amount of T cells, they're not really able to see and destroy those tumor cells. And so this is obviously a major problem. You know, it's not the same as not having any T cells, but having the wrong T cells is probably just as much of an issue. And so how do we overcome this? Well, there's different ways to do so. Our approach to do this that we've been working on for the last couple of years is to engineer T cells to do so. And the way we do that is, the thought behind that rather is that you have multiple different T cells in your body that are circulating and protecting you from different types of infections and viruses, maybe COVID, maybe the flu, and so on. They do this through their T cell receptor, as I pointed out previously. And what that means is that when they're exposed to a tumor, they really are unable to see and recognize and kill that tumor. And so why T cell engineering, what we want to do is to basically carve out that T cell receptor and replace it with a T cell receptor that's able to recognize cancer. And so now what you have is a series of T cells that, instead of recognizing all these viruses and pathogens, are recognizing cancer. And as a result, when these T cells are then re-exposed to tumors, they're able to both see and destroy the tumor. This is the basis for the work we're doing. Now, to be able to do this, you need to know which T cell receptor is actually relevant and which T cell receptor you should put into those cells. And so for this, we've developed a T cell receptor engineering pipeline, which is outlined here, where basically we take a source of T cells, whether it's blood or tumor infiltrating lymphocytes, we expose them to an antigen of interest, which leads to the expansion of the cells that we're looking for. And then we're able to enrich those cells and expand them considerably. And what this allows us to do is isolate cells that in our starting population would be potentially as low as 1 out of 70 million cells. But when we get to this point, we have millions and millions of this one T cell that's able to recognize the antigen we're interested in. And so at this point, we can then confirm that the cells are able to kill tumor cells that have the antigen we're interested in. We can then sequence to identify the T cell receptor, which is ultimately the gene we're interested in, put that gene into PBMCs or T cells that have a different T cell receptor to now have a population of T cells that are highly tuned towards killing the tumor here. And so which antigens do we go after? You're probably aware in lung cancer, there are many mutations that are called driver mutations. These are antigens, or rather, these are mutations that are found in a high proportion of patients, as is represented in this pie chart. And these are mutations that are really important because when I talk about heterogeneity, these are mutations that are unlikely to be lost because they're so important to the tumor that as the tumor changes and the tumor evolves, it's very unlikely to lose these mutations. And so we've gone after many of these mutations already for our other T cell receptor engineering efforts. And you can see here for RET, which accounts about 2.5% of metastatic lung cancer, this is what we're attempting here too. Now, when you're doing T cell receptor therapies, there's another layer of information that needs to be accounted for. It's not all to have the same potential antigen. You need to also have the same MHC molecules. Here are represented many different types of MHC molecules. You inherit one from your mom, one from your dad. And so we don't all have the same MHC molecules. And the reason this is relevant is that because of the structure of those MHC molecules, despite having the same antigens, not everyone will be able to present that antigen so that it's actually visible to the immune system. And so that's another layer of information that we account for in generating these T cell receptor engineering efforts. And here's an overview of the 10 most prevalent HLA alleles in the United States population here, which tend to be our initial set of T cells that we derive. Now, in the context of RET fusions, as you're probably aware, they don't respond very well to checkpoint blockade. Here's a waterfall plot here showing that very few patients actually responded to immunotherapy, whether it's nivolumab or pembrolizumab. Thankfully, there have been the development of selective RET inhibitors that have led to really impressive responses in a large proportion of patients. These RET inhibitors work extremely well. They don't work in all patients, and sometimes the responses are not necessarily durable. Now, when you look at these RET tumors, they're considered to be immunologically cold. And what that means here is that, for example, they have low levels of this protein called PD-L1, which is actually the target for immune checkpoint blockade. So that could be part of the reason why they don't work so well in these patient populations. But they also have what's considered a very low tumor mutational burden. And this is important because these are the mutations that are actually serving as antigens for the immune and for the T cell response. But really, you don't need a lot of antigens. You just need one good one to be able to eradicate these tumors. And so this is what we're going after for these T cell receptor engineering efforts. Now, this is the landscape of RET fusions in lung cancer. There are many different fusion partners and break sites. And so what we did was to actually cross these different RET fusion sequences to the top most prevalent HLA alleles that I showed you previously. And we came up with this list of high potential antigens for us to go after further through our experimental studies. And we performed, we input these in our generation pipeline that I showed you previously to identify T cells against these RET fusions. And so these efforts are very early on, but we're very excited to be able to show you that so far we've been able to confirm that at least 2 of these RET fusions, which are listed here, this K24R11 and C1R12, both presented on highly prevalent HLA alleles, are actually immunogenic. And the reason we can say that is because in these plots, each one of these dots represents a cell. And what we're looking for are cells that are found in this box here, because those are cells that are considered to be T cells, so the cells that we're interested in, but also cells that recognize the antigens that were listed here. And so as you can see in 2 donors for each of these antigens, we're able to confirm that these fusions are immunogenic. And so this is very encouraging. As I mentioned, these TCRs are undergoing further validation. And because we're very in the initial stages of this work, I just want to show you what the whole pipeline looks like for other T cell receptors. This is some of the work we've been doing for EGFR. You can see that, at the early stages, you have very, very few of these antigen-specific cells here that are represented in red. We're able to expand them to considerable levels here. We're able to confirm that they both see and kill tumors that have these antigens. We can sequence the T cell receptor and then engineer it into T cells that don't have it. And then we're able to purify those cells to extremely high levels, as is shown here, to confirm that in the end, when cells are engineered to have these T cell receptor, they're still able to kill tumor cells on an antigen-dependent basis, as is shown here with this blue line. And so far we made significant progress in this pipeline. This is an overview of the work we've done so far with other targets, EGFR, HER2, KRAS, some other tumor-associated antigens. You can see here the efforts that we've been working on with RET, and we validated a series of 12 different T-cell receptors so far. And just to give you an idea of the impact that this could have, here's a schematic showing the relative proportion or relative prevalence of our antigen, of the HLA allele that we're going after, and ultimately the proportion of patients that are going to be treatable with each of these T-cell receptors. And that ends up being tens of thousands of patients already in the United States with our library of T-cell receptors that we've generated so far. And we hope to be able to add some RET TCRs in the very near future. So just in wrapping this up, I want to show you guys that T-cell receptor engineering works in patients. Here are two examples of that. This is a study in pancreatic cancer where patients received a T-cell receptor against this G12D mutation of KRAS and saw the complete resolution of the nodules there. And then on the right, this is an HPV epithelial cancer clinical trial where patients were receiving a T-cell receptor against that antigen. And you can see that in all patients here, there was a shrinkage of the tumors. And actually at MD Anderson, we have an ongoing clinical trial that's being led by Dr. Negrao. And this is an example of a patient that received the KRAS TCR as well, and it led to complete resolution of the lung nodule here within only 3 months. So this works, and we're very excited to be able to participate in this going forward. And so with that, I'm going to wrap up. I just want to thank the members of the lab who contributed to this, specifically Hui, Mingying, and Peisheng, our funding agency, specifically the Happy Lungs Project, RET-positive, and Longevity. And I'm happy to take any questions at this time.
Alex, this is Dan Stromberg. Just a quick question. It's obviously a very involved, bioengineering process to get things from antigen identification, through to the point that you have T cells. What do you think the remaining timeline is for the RET work that you're doing?
So for the antigens that I showed you so far, for which we have an initial signal, I would say if, there's some technical things that can always go wrong, I would say probably within 6 months for the antigens that I showed you so far that we've confirmed to be immunogenic, assuming that all the rest pans out, we should be at a stage where we can confirm that, it's a good antigen, and the antigen is relevant in tumor cells and so on, and we have a T-cell receptor that's able to recognize that. That's for the antigens we've got so far. Obviously, we have a series of antigens, which I didn't show you, and so we're constantly kind of staggering those efforts to try to continue to screen those. But that would be for the ones that are probably the furthest along.
I see. Thank you.
I'll mention here, so Alex has got now, we've identified these TCRs, including against the RET, fusion. Once we've got the TCR, then the question is, how are we going to get it into patients? And that's an additional set of hurdles. Now, we're in discussions with Alaunos. You saw the clinical study we're running with Alaunos. That's a company that got spun out of MD Anderson, where they've got the trial up and running. We're the only site running it. We're the one place in the world doing it. And one possibility is we could bring the T-cell receptors that we identify and bring it in through that existing trial, because that trial's already got 20 different TCRs or so that are being tested for, and this would just get added to the list. And the plus side of that is we've already got the clinical study open, the manufacturing is already running, and it takes a long time to manufacture these. MD Anderson also has built their own manufacturing facility, but that would mean opening a brand new study and getting funding for it and going through the FDA for what's called an IND, which is a pretty complicated process as well. We are exploring how to do that in addition. So I think once you've got the TCR, then you figure out how do you get it into people. And we're exploring both the work with our ongoing partners versus starting a brand new clinical study ourselves.
That's amazing. Thank you.
Yeah, and I'll note that to our knowledge, these are the first RET-specific T-cell receptors ever identified. Certainly nobody's ever reported one before. So I think for the first time, we've got some hope of harnessing the immune system specifically against a RET fusion. So.
That's awesome. Thank you. All right, Dr. Heymach, do you want to present the other?
Sure, I'll fire away here. All right, so let me see. Okay, well, I'm excited we could be joined also by Ralph Kittler, who is our collaborator at UT Southwestern. Ralph, do you want to introduce yourself? I'm not sure the team has met you before here.
Yeah, so I'm Ralph Kittler. I'm faculty at the McDermott Center at UT Southwestern. I have also secondary appointments with the Simmons Cancer Center and the Department of Pharmacology. And my lab has previously developed a novel approach to rapidly identify acquired resistance mutations in cancer targets. And that is basically where I've started to collaborate with John Heymach. And we recently have also done the first experiments for RET fusions in lung cancer.
Yep. Yeah, and you'll get to see the results of this. So hot off the press from Ralph and my groups here. So just to remind everybody, so what we're trying to figure out here is when RET inhibitors stop working, why do they stop working? So you can see here, most people, if the bar goes down here, that means the tumor is shrinking. And almost everybody's tumor shrinks that has a RET fusion with selpercatinib or pralsetinib. But two things are worth noting. First of all, not every tumor shrinks equally. You can see some tumors shrink by virtually 100%, some only shrink a little bit. Here at the very bottom, this means that the tumor is shrunk by 100%. And secondly, at some point in the vast majority of cases, tumors become resistant. So what we'd like to do here is say, all right, well, instead of waiting for resistance to occur and then say, boy, how did this happen, and start our studies at that point, to say, well, let's see if we can come up with a rational prediction of what are the possible ways a given tumor could become resistant, and then come up with strategies to combat it before we're actually faced with the problem. And this is something that we've gone to school on for more than a decade with EGFR. So we've got some ideas and experience about how to do this. Now, when you talk about RET fusions, it's not one thing. So if you look at all the different ways they responded, some responded great, some didn't respond as much. We think part of the reason is because RET fusions are a lot of different things. So part of what we're doing is partnering with Guardant. And in fact, we just had a great call with them yesterday. And they've actually given us their entire database of RET fusions. And we believe this is the largest reported set. We've got about 500 right now of RET fusions. And the first thing is just cataloging all the different RET fusions. So here, what this notation means is what RET fusion means the RET gene, part of it breaks off and it fuses with another gene, and that sort of turns it on. This shows you all the different things that RET can fuse with in the database. And you can see the vast, vast majority are KIF5B RET, but there's a bunch of other things here. Now, one thing we've already seen is that these different fusions can respond differently to different drugs, and that's not surprising. They make different structures. But nobody has sort of cataloged all this. So just to ask, given a given initial mutation, what's the best drug? So you can really think of this as two parts. Given the initial mutation, what's the best drug? And then given an initial mutation and a given drug, how is the tumor likely to become resistant? And can we predict that as well? So this is really the two parts of this. Understand how different RET fusions respond to different drugs. And this might tell us we need some new drugs for different RET fusions. And then when it becomes resistant, how does a given RET fusion become resistant to a given drug? And that may be important because it may tell us, well, if it becomes resistant by one route, we can switch to a different drug that works in that particular instance, or a combination. So the first step here is a bit of brute force. And with the garden data set that we've now got, we've also got all the cases where people got multiple samples over time. So we could see when they develop a new mutation that causes resistance, what's the new mutation that happens? Now, in the literature, there's been a handful of mutations reported, but we've got a bigger set of resistance mutations now from the garden data. And this has not been, none of this has been published. So we've now engineered the most most common set of RET fusions that are out there. And you can see more than 20 of these have been made. And what each of these means, if we just pick the most common type, KIF5B RET, and this happens to be the type that Alana's got, that the two genes can break at different points and fuse together. So not all KIF5B RET fusions are the same thing. So you can see here we have listed the 1512, that's the most common breakpoint. But we've got other breakpoints here, the 2411, 2212, and so forth. And then with this, we've taken the mutations that already we know occur, and we've made those resistance mutations. So this is just saying out of all the resistance mutations that have been reported, and there's less than a dozen that have been reported. So with this data set we have here, we're more than doubling the full set of resistance mutations known to the world. But that number is still then on the order of 24 or so. So 24 is a small enough number where we can make each of those, because we can say, well, if this happened in one person, it's likely to happen again. And we can make each of these, and then we can start testing those against drugs. So this is a full set of ones we've made on the left, and a set that are in progress of being made as well. And the one mutation that's come up more commonly is this 810 mutation. This has been reported by a couple of groups in a few different patients. So that was the first one we sort of made as well. But now we've got sort of a larger set from the collaboration with Garden that we're making also. Now, most of the data in the world is based on the LC2, because this is the only widely available RET fusion cell line. But that cell line actually isn't even a KIF5B RET. It's actually a CCD6 RET. Fortunately, we now have a new cell line, MDA-L30, because we can't call it Alana's cell line and send it around everywhere. And this is a cell line we've now started sharing that we're able to study as well. So in addition to sort of the available ones, we've now got a new cell line that we can share with the world to study, as well as using a sort of blank background. We have a mouse line called BAF3, which is sort of a neutral background that we can put whatever we want in there as well. Now, from this work, we've already made some interesting observations. So first of all, we see that different RET fusions are sensitive to different drugs. So for example, here we've just made the 1512 KIF5B RET and the V3, the 2212 here. And then we've got a new cell line, the MDA-L30 with the 1512. The top ones are engineered into our sort of neutral background cell line, the BAF3s. And from this, when we test some of the drugs that are in clinical studies, we can make some observations, for example, that the 1512 seems to be more sensitive to pralsetinib and selpercatinib, much less sensitive to lenvatinib and cabozantinib and vandetanib. And what we make in our engineered cell lines turns out to match what we actually see in the MDA-L30 cell line. So in other words, what we engineer matches what we actually observe in a real patient cell line pretty closely here, which is reassuring. We also see that there's a different pattern of sensitivity with the 2212. So this already tells you if you've got a KIF5B RET with the 2212, well, you already see that ponatinib doesn't look like a particularly good drug for this, whereas pralsetinib and selpercatinib still look better, whereas ponatinib is very good for the 1512. So you can think of this now with these first couple as part of making sort of the giant library, if you will, of baseline information that if you start with this mutation, here's the drug that works best. And that's something the world doesn't have right now. And this is another example. We've got the LC2s with the CCD6, and with the CCD6 RET, we model this as well. And again, we can see a very similar pattern of sensitivity between these. So now we've built what's the world's biggest library of RET fusions, and we've started characterizing what drugs work well for this. So the plan here now is, if we make this a large enough data set, in other words, if we're able to study all these different fusion partners, different breakpoints within a given fusion partner, and then the different resistance mutations, we start to develop a big enough database where we can start grouping together what types of mutations and what structural changes are associated working with which drug. And this is the beginning of being able to functionally group together RET fusions in a useful way. Right now, everybody just gets selpercatinib or pralsetinib. Those are the only two drugs that are approved. You don't have any basis for picking one or the other. And if those stop working, you don't really have any rational basis for picking whatever drug comes next. This data set starts becoming that rational basis of which drug should you use for which fusion partner. And this follows the work we've done with EGFR, where we've identified sort of 4 structural subgroups that are now being used for clinical trials around the world. So we're thinking of the same sort of structure function based grouping now for RET that we hope becomes a standard approach. Now, the next question is, once you start inhibiting a given RET fusion, how is it likely to become resistant? And in particular, if it gets other RET mutations, so we call this a RET dependence resistance mechanism, can we predict what those RET resistance mechanisms are likely to be? Now, there's an old-fashioned approach to this, which is the following. One is wait until patients get these resistance mutations, and then make those mutations and then figure out what's going on. Now, we're doing this old-fashioned approach, but we're limited there. Right now, the whole world only knows about 20 of these mutations, even though we know thousands of people have had RET fusions and been treated and become resistant. So the world is not very good at reporting what resistance mutations emerge. And that's because, in part, people don't typically even look for resistance. They don't have anything to do with it. So the question is, can we come up with a quicker way to identify the resistance mutations? Now, the other way we can do it is in cell lines or in mice. We can take lots of cells and treat it with the drug and start with low levels of the drug and keep escalating and escalating. And then sometimes after a year or 2 years, we might get a resistance mutation or two, and then we can figure out what those resistance mutations or two are. But in that, it may take a year to get 1 or 2 resistance mutations, and that doesn't tell us, have we looked at all possible ways the tumor can become resistant? So this is why we've partnered with Ralph to say, can we come up with sort of an accelerated evolution to rapidly look at all the possible resistance mechanisms that can occur? And then we can sort of overlap that with what's actually occurring in patients. So let me try to illustrate this. So again, these are all the resistance mutations that have ever been reported. Even though there are thousands of people who've become resistant, these are the only ones we know about, the only resistance mechanisms. So the way I'd like to explain this approach is starting with the Galapagos finches. And so bear with me for a minute. This will hopefully make some sense, shortly. You probably all know the observations that led to the key breakthrough with the theory of evolution, of course, was the observation on the Galapagos Island that all these finches that started off, from a single finch, developed different beaks. And they could sort of track that the different beaks let them, find birds and find bugs in different niches or eat different types of things. So essentially, these different beaks allowed them to explore different evolutionary niches. Now, let's say I gave you a different problem. Like, let's say we had a brand new space, a new island, and we said, hey, can we predict what type of finch is most likely to survive on this island? Well, you could sit there and wait 100,000 years and just see which bird ends up surviving. But a different approach would be, well, if you could collect every finch that ever lived and put them all together there at once, and then let them compete. Well, this is certainly going to be a lot quicker than waiting for evolution to run its random course over hundreds of thousands of years. So you're putting every potential finch out there and letting them all directly compete at one time to see which one is most successful in a given new island. So this is a little bit what the lentimutate system does. So what lentimutate does, and this technology that Rolf has helped pioneer, is we take a RET fusion, but then you use a lentivirus that is error prone. This means whenever it divides, it makes lots of mistakes. And those mistakes occur sort of randomly over a given area. So we can induce millions of different mutations in RET at one time. And they're all present in the very beginning. From the first moment we start the experiment, we're starting off with 1 million different mutations. And then we start treating with the drug and we say, all right, you 1 million mutations, compete and see which ones survive. And this then tells you which mutations provide fitness, essentially will enable a given RET fusion to survive against selpercatinib or pralsetinib. And then we can see, do different drugs have different patterns of which mutations survive? So to give you an idea now, we actually have the first results back from this. And the first thing you'd like to know is, well, does this give you results that are clinically relevant? So here, Rolf's group has already completed now, we've given him the KIF5B, the 1512 fusion. And we've gone through with selpercatinib as well as pralsetinib. And the first question is, do you get anything that's clinically relevant? So what we're looking here now is how many fold enrichments, so what mutations survived. So Rolf takes how many were present in the initial mixture, and then whichever ones survive after a given period of the drug, and survive and divide and sort of thrive. And then what's that ratio of enrichment? So for example, these top mutations, you can see 300-fold enrichment. That means they're present at a 300-fold higher rate than they were initially. And Rolf, do you want to give them an idea what's the initial starting size, the number of mutations that are induced here when we're starting?
So that's dependent on the initial cell population. So for these experiments, we started with about 10 million cells, and we assume we have an average mutation rate of 1 in 3,000. So we will have probably, we assume that the gene has approximately 3,000 nucleotides. Yeah. So we would probably have a few million initial variants. Right. Yeah, that we now selected from.
Right. And so what we actually see here, and if you look at these mutations, so what are the top 2 mutations? So the top 2 are the V804M or E and the G810S. Now, nobody told Rolf or his cells 810 and 804 are the 2 mutations that have already been observed. But Rolf was able to detect this in the time course of less than a month from the time we gave him the fusion. It took the entire field more than 5 years to come up with this set of mutations that we found in less than a month from the time we started these experiments. So that's the first interesting observation. So it gives you an idea of the accelerated time frame that we're able to detect resistance mutations. Now, the second interesting observation is that the mutations that cause resistance to selpercatinib are different than the mutations that cause resistance to pralsetinib. Now you see a couple of them are the same, right? So you see the 810 here and you see the 810 here. So the 810 mutation can cause resistance to both, but the 804 causes resistance to selpercatinib. We didn't see the 804 with pralsetinib. You can see, so you can see the different distributions. You can see the M918T. And by the way, the M918T has also been described as a resistance mutation to selpercatinib. It hasn't been observed for pralsetinib. So from day one, you know, if we have somebody treated with selpercatinib who comes along and, you know, we find out, you know, that they happen to have an 804 mutation, we'd say, well, actually pralsetinib looks like that may be a very good drug. You know, likewise, if somebody comes along treated with pralsetinib with the 730I mutation, you can say, well, that does cause resistance to pralsetinib. It doesn't seem to cause it to selpercatinib. Now, what we're in the process of doing now is taking these mutations and engineering them into the specific construct, the KIF5B 1512 RET, and then engineering these specific mutations. We've already made the 810s and we're making the 804s. We're going to make the others. And then we can go back and specifically validate which drugs work or don't work. But once we've made these resistance mutations, we can actually test, you know, the turning point drug and cabozantinib and vandetanib and all the drugs that are out there in the world. So if somebody shows up with this mutation, we can say, listen, we already asked this question and here are the 5 drugs that work and the 3 drugs that don't work for this resistance mutation here. So this is just the progress over the last month. And again, I think we've got more data about resistance mutations to these different drugs than sort of exists in the world publicly right now. We're also looking at RET independent ways about different pathways that can get turned on outside of RET here. And with this, we've been doing an approach called RPPA, reverse-phase proteomic arrays. And so we've already got a robust set of targets that we can go after, you know, here. And VEGF, EGFR, and mTOR are 3 of the pathways that have emerged from this. So we're in the process of studying some of these different combinations already. So you can see here data with vandetanib and vandetanib plus an mTOR inhibitor. We're able to overcome some of the resistance here. So we now are really sort of thinking about 2 approaches. If the resistance is caused because you get another RET mutation, we're figuring out, is there a different RET inhibitor that could work? And then the second is if it's resistance because you're turning on a different pathway, can we find a combination of drugs that works? Now, it's possible as we go through this that there are some mutations that none of the drugs work for. So for example, the 810 mutation, so far pralsetinib and selpercatinib, neither one works. We don't know if any of the other drugs work. If they don't, what we may set about doing is saying, oh, well, can we make a drug that does work for that? You know, and then we can engineer it with a very specific goal in mind, you know, to engineer it for 810 specifically there as well. And finally, and again, there's a lot of work led by Monique in our group, but looking at these cells that are left over after you've killed most of the cells with the RET inhibitor. Usually we can kill 95 to 99% of the cells. Well, those few cells that are left over, we call drug-tolerant persisters. And what we're trying to figure out is what gets turned on in those drug-tolerant persisters that lets them survive. We've already got an interesting target here called CD70, and it's something we had learned from EGFR earlier. Now we see with RET inhibitors, CD70 gets turned on. And the interesting thing is we've got a few different ways to target CD70. We're making CAR Ts. We're making some new antibody drug conjugates. So this may be another approach, is kill as much as you can with the initial RET inhibitor and then use a CD70 targeting agent to try to get everything that's left over there as well. So that's the approach. You can see we're taking a few different streams here, but already between the lentimutate and the BAF3s, I think we've characterized to a greater extent than has been done before all the different types of RET fusions and different ways they can become resistant. So we're well on our way here.
Thank you, John. That's amazing. I'm now going to turn it over to Joel Feinberg, who's chairman of the board for the Happy Lungs Project, to just do some ending remarks. Do we have time for, no, we don't really have time for Q&A. If anybody has any questions, feel free to send them to happylungsproject.org and we'll get them answered for you. info@happylungsproject.org. Joel, you're up.
Thanks a lot. First of all, thanks to Dr. Reuben and Heymach for both presentation and all the time and effort in trying to solve this problem. our family back more than about a year and a half ago started the Happy Lungs Project at first because Alana was diagnosed and we were desperate to find a cure and reliable treatments for people with RET-positive lung cancer. So we embarked upon this venture to solve what we thought was a solvable problem. And in the course of that, formed this foundation that was to spur and understand and bring together the best researchers, collaborators, scientists, clinicians to figure out how to address RET-positive lung cancer. And while the RET-positive lung cancer may be a relatively small portion of all RET-positive lung cancers, all of the research we do, we believe are going to help the 120,000-ish thousand people a year that are affected by RET-positive cancers. So the treatments that we're discovering here and the work that's being done at MD Anderson as well as at Stanford and Mass General and Sloan Kettering are all to benefit what we believe to be just such an important cause. So the research that Dr. Reuben addressed is some of the research that our family funded initially when we first started the foundation. And as you can see, that's building on some very important foundational work that we plan to expand on and make a huge difference. So the money that both we've given personally and the money that we've raised and the money that, quite frankly, we hope to raise from people who are on the call or people who know people who are on this call and in the community is to help fund this very important research that is literally going to make the difference of people surviving this or not. And we are determined to find a cure. And our goal is to raise $10 million over the next couple of years to fund all of the necessary research to find an answer. So with that, if there are questions that we can answer, if there's people who want to get involved, we welcome that. If there are, if people are inclined to donate, we would encourage and welcome and appreciate it because the work we're doing is incredibly important and will in fact save lives. And that's the purpose of this organization.
Thank you, Joel, and thank you everybody
Sure.
for joining us today. We could not be doing this without each and every one of you. And we'll keep on until we find a cure.
All right.
Thanks, everyone.
Well, thank you to Happy Lungs. We really appreciate the support.
Of course, we appreciate your work. Thank you, Dr. Reuben and Dr. Heymach and Dr. Kittler,
Yes.
and all the others who are working so hard on this and making this your priority. You are our heroes.
Great.
Happy holidays, everybody. Goodbye.
Thank you very much.
Oh, thank you.